Welcome to our conversation on molecular biology. These short lectures are going to outline the structure of DNA. We'll discuss DNA replication, which is what our cells do right before they undergo mitosis. They have to duplicate DNA, and so we'll go through the process of how that happens in more detail. We're also going to discuss transcription, translation, and how individual genes on a chromosome actually produce products like proteins and things like that. The first part of this discussion will be about the structure of DNA and DNA replication. When we look at a chromosome, like on the bottom of this diagram, what we're seeing is a condensed collection of chromatin. It's a condensed collection of the genetic information that is on one chromosome. So on the very bottom is a duplicated chromosome. Remember, these are two copies and they're attached at that centromere. But if we look closely at that chromosome, at that arm of the chromosome, we can see that it's made of condensed chromatin. As we move up this diagram, we can see that it's made of this wound up chromatin. As we go to the center of this diagram, we can see that that chromatin is coiled around these histone proteins. And then if we go towards the top, we can see that that coiled chromatin is made up of one strand of DNA. And that DNA consists of that double helix shape that you're familiar with. Now here's a very close-up image of that double helix shape. And we can see that there's a couple of components that contribute to that shape, namely 4 different kinds of nucleotides: thymine, cytosine, guanine, and adenine. The nucleic acids are a group of important macromolecules. These are macromolecules we discussed in earlier labs, and we're going to revisit this macromolecule and go into more detail about it. Nucleic acids are made up of a couple of different parts. On the very left, you can see the basic structure of a nucleotide. That structure, in this instance, is made of 3 different colors. There's a yellow phosphate group, there's a 5-carbon sugar, and that sugar is different depending if we're talking about DNA or RNA. Let's look at the 5-carbon sugar first. We can see that in the case of RNA, that sugar is ribose. In the case of DNA, that sugar is deoxyribose. The difference between ribose found in RNA and the deoxyribose found in DNA is that ribose has a hydroxyl group at the 2' carbon. The nitrogenous bases are split into 2 groups, the pyrimidines and the purines. The purines are larger than the pyrimidines. The pyrimidines include cytosine, uracil in RNA, thymine in DNA. The purines include guanine and adenine. Here you can see that thymine and adenine are bonded by 2 hydrogen bonds. Below that, guanine and cytosine are bonded by 3 bonds. This means that whenever you find thymine, it has to be complemented by adenine. Or if you find guanine, it must be complemented by cytosine and vice versa. On the left of this diagram, you can see that adenine, thymine, guanine, and cytosine are each represented by a different color bar with the shape at the end that's complementary to one other nitrogenous base. The round end of adenine is complementary to the end of thymine, and the pointed end of guanine is complementary to the end of cytosine. This is to help visualize how these different nitrogenous bases will relate to each other within the double helix structure. On the far left, we can see a double helix structure, and you can see those nitrogenous bases represented by colored bars. The base pairs can only match up with complementary base pairs, which is dictated by those hydrogen bonds on the right. Also helpful to note in this image is that there's a 3' and a 5' end to each strand on the double helix of DNA. Here's a comparison between RNA and DNA. There's 2 things to take note here. One is that RNA is a single-stranded molecule. It's not a double-stranded double helix, as in DNA on the right. Also, the nitrogenous base thymine in DNA is absent in RNA, and instead we find uracil. Uracil is the complement to adenine in RNA. The genetic information within a eukaryotic cell is contained within the nucleus. There you'll find chromatin, which is individual fibers of DNA. Prokaryotes typically contain RNA in a ring. That ring is found in a condensed area within the prokaryote called a nucleoid.